So thank you to the organisers and ABCAM for actually arranging the symposium and giving us a chance to look backwards, to look forwards, and to just think about where the science is going and the areas that we're really excited about. And this is a picture I really like because it really, one, is very beautiful, and two, it illustrates for me a really interesting point about epigenetics and the regulation of gene expression. So if you think about a caterpillar and a butterfly, they have exactly the same genomes. But by very carefully and very precisely regulating which genes they express, you can actually produce totally distinct forms, totally distinct functions, and absolutely different physiology. So if you think about the power of the ability to control gene expression, epigenetics becomes really important. And what we've learned over the last few years is, contrary to our expectations, epigenetics isn't fixed during embryonic development. So when a neuron forms or a skin cell forms, we'd always thought about epigenetics as being really important in deciding cell lineage and cell fate. What's been really interesting and exciting over the last few years is that we've learned that epigenetics continues to be important throughout life. It responds to the environment and it changes in disease. And very importantly for us, it is actually amenable to the use of small molecules to alter epigenetic function to potentially reverse or impact on disease progression. And so if you think about genes and the way they function and how the different states they can be in, and this is work taken from Ruslan Metitov and a number of his colleagues over a number of years, if you look at, say, gene expression in the immune system, you can think of genes in a number of different states. You have those genes where the DNA is tightly wrapped around the chromatin. It's in a locked position, transcription factors can't get to it. That gene is stably repressed and is off. And the example there would be, say, the hemoglobin gene in dendritic cells. You don't need to express that in those cells. At the opposite end of the spectrum, you've got genes that every cell needs to express, and things like GAPDH, where the chromatin is open, and the histone proteins that the DNA is wrapped around have already got the marks on it to say, express this gene, keep it on, make it at high levels. And the example there that I give is GAPDH in dendritic cells. Then you've got other sets of genes where you actually want to respond very quickly. So in response to a signal, you want to be able to turn on these genes within the matter of minutes. And those are genes that, say, allow you in the immune system to respond to a bacterial or a viral infection. So these genes are poised, the DNA is open, some of the marks on those histone tails are already there to say, in the event of a stimulus, turn this gene on very rapidly. And the examples there would be things like interferon beta in plasmacytoid or dendritic cells. These are cells that respond to a viral infection very rapidly. Then you've got another set of genes which turns out to be particularly interesting, and I'll come back to these in a while again, and these are called secondary response genes. These are genes that encode factors that, when they're made, actually produce dramatic biological effects. You want to control the expression of these genes very tightly, and these secondary response genes actually require two things to happen. You require remodeling, so you need to evict these histones, you need these factors to come in, and you need those factors to now start writing these marks on those histone tails to say, turn this gene on. And these genes then require the interaction of a whole set of complexes to turn those genes on. And the example there would be something like IL-6 or interferon beta in macrophages. If you overexpress IL-6, it's associated with a lot of autoimmune diseases, so you really want to regulate that. And this kind of illustrates to us the importance of very precisely controlled gene expression. And the immune system is one system that allows us to explore that very rapidly and very efficiently, because unlike cancer cells, you haven't got a lot of conflicting mutations and a lot of background of many changes across the genome. These cells can be precisely formed, can be precisely stimulated, and you can look at these responses in vitro and in vivo. So if you really think about the immune system, it's absolutely perfect for epigenetics. So every cell in the immune system needs to know who it is, where it is, what it's seen, and what it's going to need to see. And if you think about that, that's going to be epigenetically imprinted. And you can see that from a lot of different people's work, it's actually become very apparent that at each of these cell decision points, there's a complex interaction between transcription factors, epigenetic machinery, and chromatin remodelers to allow these stable memory decisions to be made. One of the other things that we've learned is that much of the variation that's associated with disease actually doesn't happen within coding regions. As we've started to be able to map enhancers and distal regulatory regions, and this is work done by Morano and colleagues, where they took genome-wide association studies and said, where is the variation happening that's associated with predisposition to particular diseases? More than 60% of it was actually happening in enhancers very far away from the actual genes. And what you see is that much of that variation actually now starts to fall into these enhancer regions, which are only active, which are only open in particular cell types. So they're cell type specific regulatory regions, suggesting that immune cell identity is epigenetic, and often immune cell dysfunction is likely also to be epigenetic as well. So there are many, many different mechanisms associated with epigenetic regulation of gene expression, and they range from everything from modification of the DNA bases themselves, like cytosine methylation. They involve non-coding RNAs, but the area that we've been focused on is really the post-translational modification of these histone tails. So when DNA is tightly wrapped around the histones, these tails stick out and they can be chemically modified with tags, which can either be acetylation, phosphorylation, methylation, etc. Those chemical tags essentially function to form mechanisms to allow the marking of sets of genes to essentially tell them when they should be on, when they should be repressed, or when they need to be expressed in response to particular stimuli. And those marks are added by what we call the writers. They're removed by sets of enzymes that we call the erasers, and they're read by proteins that recognize those marks and then assemble particular transcriptional complexes in response to the stimuli that I mentioned before. The really interesting thing in the whole the area of epigenetics is the degree of progress that's been made in identifying and progressing molecules into clinical development. And you know that there are pan-DNMT inhibitors that are licensed for the treatment of a number of different leukemias. These are non-selective DNMT1, 2, and 3 inhibitors. Very recently there's been the exciting approval of the IDH2 selective inhibitor for the treatment of different leukemias. And you'll know that there are a number of pan-HDAC inhibitors that are approved, again, for the treatment of different myeloid leukemias and lymphomas. So that first generation of molecules were characterized by being non-selective pan-DNMT or pan-HDAC inhibitors. What we're starting to see, and we've been involved with this, is actually the generation of the second generation of epigenetic inhibitors, which show greater selectivity and therefore associated with better tolerability. And one of the most dramatic observations and learnings that we've had over the last few years is, almost as you'd predict, as we make more selective inhibitors, the sets of genes that you regulate are much more defined. And many people's expectations on epigenetics have been based actually on understanding of these non-selective inhibitors. And what we're seeing with these new inhibitors is, as we make them better, as we target them better, as we make them more selective, we're actually starting to see very precise sets of genes being modulated. And GSK and Horizon have taken the first LSD1 inhibitors into the clinic. Epitherapeutics have generated the first inhibitors of the JARIDs. We generated the first Jumanji D3 UTX inhibitors, which show very nice and selective modulation in the immune system. We've actually been able to use this targeting technology that allows us to deliver HDAC inhibitors just into monocytes and macrophages. That's allowed us to take a pan-HDAC into patients with rheumatoid arthritis, and we generated the first ever PAD4 selective inhibitors that we've partnered with BMS via Padlock. In terms of the writers, Epizyme have made some great progress in taking the first DOT1L inhibitors into the clinic. GSK, Epizyme and Constellation have taken the first EZH2 inhibitors into the clinic, and there's some really impressive efficacy data being reported in patients with mutant follicular lymphomas. And GSK has taken the first PRMT5 inhibitor, and in the next few weeks you're going to hear some news about another PRMT inhibitor that's just about to move into the clinic. In terms of the readers, I'll talk a little bit about some of the work that we've done with BET, but there's also been a whole range of different reader inhibitors that have been generated, started to be characterized, and again are showing really surprisingly selective effects on gene transcription. So I've talked a bit about the readers. So lysine residues on histone tails can be post-translationally modified, and if we think about the acetyl code, those acetylation marks are added by histone acetyltransferases. They're removed by the histone deacetylases, and those post-translational marks are read by these bromodomain-containing proteins. Contrary to everybody's expectations, we were actually able to develop and identify very selective BET bromodomain inhibitors that just inhibited BRD2, 3, and 4, and T actually had no effect across the whole family of bromodomain-containing proteins, which many people had not anticipated, given that they were just recognizing a very simple post-translational modification. We were able to show that those inhibitors bound into the binding pocket where the acetyl-lysine residue would normally sit and actually function as a competitive antagonist. And the result of that, actually, we've characterized in cancer cells and taken the first of those inhibitors into phase 2 studies in breast cancer and prostate cancer, and we'll come back and talk about the data in the next few years. I'm going to talk today more about the work we've been doing, as I mentioned, in the immune system using these, and we've been able to show that BET proteins turn out to be really important in modulating immune cell dysfunction, and we've shown this and other people have now confirmed this. I'm going to talk a bit about the work we've done in macrophages. So working with Sasha Tarakovsky at the Rockefeller, we take mouse bone marrow-derived macrophages, differentiate them in vitro and treat them with LPS, which is a TLR4 agonist, which makes those cells think there's an infection going on. They turn on about 800 genes after 4 hours, and to our surprise, what we saw was that actually only 150 of those genes were regulated by the presence of the compound. If you look to see where the BET proteins are across the genome, it's present on thousands of genes. And the knockouts had told us that it was going to affect thousands of genes, and everybody's expectation was that inhibiting these proteins would have global catastrophic effects on transcription. We were really surprised to see a really select set of genes being modulated by these inhibitors, and we were able to show at the promoters of those genes that those compounds were preventing the recruitment of these proteins to those enhancers and promoters, and that inhibition was correlating with our ability to regulate that gene expression. It turns out that that subset of genes that we're affecting actually turn out to be really important. If you give a high enough dose of LPS to mice, you induce a sepsis-like syndrome. Those mice develop a massive cytokine storm, their temperature drops, the fur ruffles, and then within about 24 to 48 hours, all of those mice are dead. What we found was that a single dose of the IBET compound, given either 90 minutes before LPS, or even more remarkably, 90 minutes after LPS, after the primary response genes had already started to be induced, we were able to keep all of those mice alive, suggesting that the subset we were affecting was actually a really physiologically important set of genes. So what were they? So if you look at the genes that are induced, what we saw, as I mentioned earlier, where you see all these primary response genes come on very rapidly in response to LPS, none of those were affected by the presence of the compound. In the same cells, the same concentration of compound was selectively affecting 95% of these secondary response genes. And those secondary response genes turn out to be really important because many of these that we know from studies in the immune system turn out to be really important for driving autoimmunity and pathological immune cell function. So we'd actually accidentally discovered that inhibitors of BET proteins can modulate sets of genes that turn out to be important in the immune system. And now you can actually do some reverse translation. You can actually now take some of these molecules that we've developed to take into the clinic, to start to explore fundamental aspects of biology? Can you use these now to start to dissect aspects of gene transcription? So again, now looking at macrophages, if you stimulate them with this TLR4 ligand, you can actually look genome-wide to see all of the enhancers that are being turned on in response to that stimulation. And what you see is that this mark of this dimethyl mark is being added as those enhancers are being stabilized and formed. There are a number of genes that are being turned off where that mark disappears. And what you see, what we found was that actually that enhancer formation and stabilization actually required transcription of those enhancers. If we block the transcription with these BET inhibitors, you could actually stop these enhancers being formed. There was a beautiful correlation of the enhancer RNA expression and the distal genes they were regulating, suggesting that these distal enhancers were actually coordinating transcription at the enhancer and the gene body. And that inhibition of transcription and elongation prevents enhancer stabilization, so you can start to explore new aspects of biology. As you'd hope, if we then go into in vivo models of immune cell activation, if we take an IBET compound and put it into a rat collagen-induced arthritis model, what you do is you inject an adjuvant plus collagen protein, you activate the immune system, and the rat's immune system now starts attacking the joints. You get inflammation in the joints and bone damage. You can then measure that clinical score. And what we found was actually just treating the compound for the first 10 days was actually able to epigenetically reset, and those rats never go on to develop disease. We also found that if you waited 10 days and then started treatment once the immune activation had already started, that was also able to completely reverse the development of arthritis. And it also worked in a different model of arthritis where you do a serum transfer model. Consistent with that exciting preclinical biology, I'm pleased to say we've now been able to take a BET inhibitor that we can target to monocytes and macrophages. So in addition to the molecules we've got progressing in oncology studies, we've been able to take the first BET inhibitors into patients with rheumatoid arthritis, psoriasis and lupus using this targeting technology, and we'll be able to come back in a few years and tell you about the progress in that space there. So I hope I've been able to share with you some of the excitement and how epigenetics, which started in cancer, where cancer cells forget who they are and when to stop dividing, to the immune system, where immune cells who normally need to know who they are and what they need to fight actually forget what self is versus foreign is, and how actually that's a huge new frontier for the application of epigenetics. Can we use epigenetics to retrain the immune system to actually fight autoimmunity and inflammatory diseases? And we're starting to see actually probably the next frontiers are going to be metabolic disease and neuroscience. Again, cells that need to remember who they are and where they are and just remember. And those are the areas that we think epigenetics is moving into. We think epigenetic mechanisms are going to be important in allowing the very precise control of gene expression. I've shown how some of these epigenetic inhibitors, as well as being able to progress into the clinic, actually allow us to identify exciting therapeutic opportunities for modulating human biology. Those compounds actually allow us to dissect new areas of biology to start to understand epigenetic mechanisms at the cell, the gene, and the tissue level. And as Alan mentioned at the beginning, since we started our epigenetics effort in 2012, we've now been able to advance 7 different epigenetic molecules into clinical development. And that is quite exciting for a whole new area of science to actually translate fundamental scientific research into clinical exploration. And with that, I will stop and thank you.